Okay, so for blood one, we're going to talk about the 8 functions of blood, describe the composition and physical characteristics, functions of whole blood, explain why it's classified as a connective tissue, discuss the composition and functions of plasma, describe the structure, function, and production of erythrocytes, talk about the chemical composition of hemoglobin, and we're going to give examples of disorders caused by abnormalities of erythrocytes and what goes wrong in each disorder. So I already mentioned it, but what blood is a tissue from which 4 of our main tissue types? Connective tissue. So we're actually not going to discuss really the functions of blood. If you look at your lecture notes, there's a whole section about like transport and such. Just read those functions on your own, learn those on your own after class, because they're just lists of functions, and I want to make sure we get done with lecture at the end of lecture today. So the first thing we're going to talk about are fluid compartments. So our body's water is divided between 3 fluid compartments. And this isn't in your lecture notes, but you can make some just short notes on the side about this, or actually the last page of the lecture notes is blank on the other side, so you should be able to do it there. But the body's water is divided between 3 fluid compartments. So our total body water is about 60% of our body weight. 40% of that 60% is going to be, of that water is within intracellular fluid. So 40% of our body weight is just the fluid that's inside all of our billions of cells. One thing you should know, and how many of you are taking or have taken medical terminology? So a few of you, but some things you can jot down to kind of help you remember or understand words as we go through. Intra means within, while inter means between. So when we say intracellular fluid, that is the fluid within or inside the cell. And then we have our extracellular fluid, which is the other 20% of that 60. And extra means outside. So this is fluid that's outside the cell. And that can be further divided into 2 subcompartments. We have the interstitial fluid. So this is and inter means between, right? So interstitial fluid is the fluid between our cells and our blood, right? And it says specifically plasma because plasma is the fluid part of our blood. And that plasma is also referred to as our intravascular fluid. So that means within our vessels or vasculature. So we have our intracellular fluid inside the cell, then we have our extracellular fluid, which is outside the cell, but the fluid on the outside of the cell is divided between 2 other compartments, which is the interstitial fluid, all the fluid in between our cells. Does anybody remember what the I don't want to say the word, but what the fluid is between cells when we learned about tissue? So epithelial cells has little to no of this, and connective has lots of it. Yeah, there's another name for it. The extracellular matrix. Remember learning about the extracellular matrix? So that's the fluid that we're talking about. So that's the interstitial fluid. And then again, our plasma is our intravascular fluid. Okay, is everybody okay with those 3 fluid compartments? Okay. Anybody still need these notes up? Okay. Okay, everybody good here now? Okay. And so the diagram that you're seeing here is showing centrifugation So centrifuging, it involves putting tubes inside of a centrifugation machine, and what you're going to do is you're going to spin that at very high speeds, and it's going to separate the fluid by density. And so when we talk about the different components of blood, we have our plasma, which is going to be this top yellow layer, and then we have our buffy coat, which is this almost translucent layer in the middle. And then our erythrocytes, or our red blood cells, are at the bottom. So our erythrocytes are the most dense part of blood. And so when we spin it, it's going to be the one that's at the bottom. Our plasma is mostly fluid with some solutes. So that's the least dense, and it's going to be at the top of those layers. So within your notes, it describes those three components of blood. So plasma is about 55% of our whole blood. Again, it's the least dense component, which is why it goes to the top when we spin it. It is going to be a straw yellow color. It is over 90% water, so mostly water. We just talked about how it's one of our three main fluid compartments. And then it's going to contain dissolved solutes. So hormones will be dissolved in our plasma. Glucose will be dissolved in our plasma. Some blood proteins that we're going to talk about are going to be dissolved in our plasma. Ions. All the things that our body needs to have available are going to be dissolved in the plasma. Then we have what are called the formed elements. So these formed elements used to be called, and they're no longer called this anymore, but they used to be called cellular elements. Cellular elements. And the reason why they were called cellular elements is because it contains our leukocytes, which are our white blood cells, our platelets, and then our erythrocytes, which are our red blood cells. So we used to call these cellular elements, but we don't anymore because some of these components here, even though they're called like erythrocytes, an erythrocyte is not a true cell. It's missing lots of different components that we'll talk about in a second. And then platelets are only a fragment of a cell. They're not even a whole cell. So we've changed the name from cellular elements to now formed elements because not all of these components are true cells. So we'll talk about the buffy coat first. So buffy coat has our leukocytes. I know in BIOL 32 you were allowed to call them white blood cells. You cannot call them that anymore. They are leukocytes. And then our platelets. Platelets, again, are not true cells because they are fragments of a larger cell called a megakaryocyte. Okay, so platelets are formed from this very large cell that we'll talk about, I don't think today, I think we're going to talk about it on Monday. But it's fragments of this very large cell called the megakaryocyte. And we kind of just break off pieces of that larger cell to make our platelets. And so those two things are leukocytes and our platelets. And also, you can call them platelets, but the scientific term for platelets are thrombocytes. So if there is a question on like the practical that asks for platelets and you remember to write thrombocyte and you spell it correctly, then I'll give you an extra quarter of a point if you so want it. But thrombocytes. So that's our buffy coat. It's almost a translucent layer that's in the middle of our three layers of blood. And then last is our erythrocytes. Erythrocytes are about 45% of our whole blood, which is what we call our hematocrit. And we'll talk about that in a couple of slides, and it's the most dense component. And we'll talk about a little bit later why they're not true cells. But I guess I'll just tell you now. Erythrocytes don't have a nucleus, don't have mitochondria, and don't have a lot of the other organelles that other cells do. So it's not really a true cell. It's missing lots of components. Okay, so questions about the different components of blood. Okay. So here are three fluid compartments again. We have our intracellular fluid, which is within the cell. We have our plasma, which is within our blood vessels or intravascular. And then we have this interstitial space or interstitial fluid that's in between those two other compartments. And so, can anybody tell me what the barrier is between the intracellular space and the interstitial space? What's keeping those two separate from each other? The plasma membrane, yeah. So this would be our plasma membrane. And then what's the barrier or wall between the interstitial space and our plasma? What is it? It's going to be our capillary wall. So the wall that's enclosing our blood is going to be our barrier between the interstitial space and the plasma. So this will be our capillary wall. And I say specifically capillary because at the capillary is where we can exchange fluids between or solutes between these compartments. You can't do it at arteries or veins because they're too thick. The walls are too thick and you can't share between. Capillaries is where we're going to be exchanging fluid and solutes. So you do have to know the concentrations of certain things in these different fluid compartments. You know a little bit about it already because of action potentials and membrane potential that you learned about in BIOL 32. If you remember, we have a little bit of sodium inside of our cells and lots of sodium on the outside of our cells. This allows for depolarization of our cells. If you remember, sodium slowly leaks in to our cells, our neurons and muscle cells, in order to get that membrane potential to increase and increase until it reaches threshold, and then we can send out an action potential. And so we have lots of sodium on the outside of our cells and a little bit on the inside of our cells to allow for that depolarization, to allow the sodium to go from an area of high concentration to an area of low concentration to reach threshold, send an action potential, and get some kind of response to happen. So in 32, all you needed to know was a lot on the outside and a little bit on the inside. Well, now you need to know exact concentrations. So on the inside of the cell, sodium is going to be 12 milliequivalents per liter. And then outside the cell in the interstitial space is 140 milliequivalents per liter. And then in the plasma, it's also going to be 140 milliequivalents per liter. Now remember, both of these spaces together, the interstitial space and the blood plasma, both of those together are considered the extracellular space. So you're going to notice as we move on throughout the semester and in this diagram here as well, that our interstitial space and our plasma are going to have equal concentrations of a lot of different things. Okay, they're both considered outside the cell, and they keep pretty equal concentrations between those two. One way that we keep those two equal at 140 milliequivalents per liter, even though solutes are moving in and out and moving all the time, is that we use a protein channel here to allow for the movement of sodium in between or between those two fluid compartments so we can keep those concentrations equal, 140 on one side and 140 on the other side. Does anybody remember the type of transport that allows us to reach an equilibrium, but we have to use a protein channel to do it? Not active. What is it? Facilitated diffusion. So with facilitated diffusion, we go from areas of high concentration to areas of low concentration to reach an equilibrium. But when we do it, we need a protein to make that happen. What is it called when we don't need a protein at all? Simple diffusion. Yeah. Now, you may have not heard of this unit before, milliequivalents per liter. And there are some notes right below this diagram in your lecture notes. So units are measured in milliequivalents of solutes per liter. This is how we measure electrolyte concentration specifically. So we'll talk about milliequivalents with sodium, we'll do it with potassium, we'll do it with calcium. So only electrolytes only, those things with a charge. And why we use milliequivalents per liter is because it's related to its electrical charge. So how we come up with that number is by multiplying the ion concentration by the charge. And we'll talk more about how electrolytes manage our fluid balance when we get to the end of the semester. The next solute we're going to talk about is potassium. Potassium, if you remember, we have a lot of it inside the cell, a little bit on the outside of the cell. So after we've done our depolarization and we reach that peak of the action potential, what is it called when we come down in that membrane potential? Repolarization. And repolarization is caused by the efflux or the movement out of potassium, which is why we have lots on the outside and a little bit on the lot on the inside and a little bit on the outside to allow for that movement out and allow for that efflux so we can have that repolarization. So it's really, really important in this class that you continually brush up on your BIOL 32 information because we talk about it a lot. So the exact concentrations are going to be 140 milliequivalents per liter on the inside of the cell. So lots on the inside, not so much on the outside to allow for an efflux during action potentials. On the outside, it's actually only 4 milliequivalents per liter. And like I said, because both the interstitial space and the plasma are considered extracellular or outside the cell, they usually have equal concentrations of solutes in these two. And this is also carried out by a protein channel via facilitated diffusion. And remember, the only things that can move across a membrane by simple diffusions are things that are small, nonpolar, and uncharged, right? And so, or water, or fat soluble and uncharged. And so the reason why we need that protein channel is because the sodium and potassium, they're charged. So they wouldn't be able to cross a membrane on their own with a charge. So they need that protein. They need the facilitated diffusion to allow that movement between those two spaces. Okay. Now, one thing that we haven't mentioned yet, okay, so we talked about how sodium is a lot on the outside of the cell, a little bit on the inside. Potassium is the opposite, a lot on the inside, and a lot on the inside, yeah, and a little bit on the outside. And so we have uneven concentrations across the membrane here. And so what is the protein that allows us to keep that uneven concentration? What do we call that? That moves sodium in one direction and potassium in the other direction. The pump, yeah, the sodium-potassium pump. So this guy right here represents our sodium-potassium pump. And it's what it is what allows us to keep sodium high on the outside and keep potassium high on the inside. This is the sodium-potassium pump. And what kind of transport does it have to use in order to keep those uneven concentrations? Active transport. And active transport is going to require what? ATP. And then the last solute that we're going to talk about is protein. Now, protein is how these two extracellular spaces differ, because we are, of course, going to have protein on the inside of the cell, because what organelle do we have on the inside of the cell that's constantly making protein? It's ribosomes. So ribosomes are constantly making protein on the inside of the cell. They read that mRNA to bring the amino acids and with that tRNA to make the protein. And so we're always going to have lots of protein on the inside of the cell. We're always also going to have proteins on the inside of our plasma in our blood, because our liver makes blood proteins for us that I'll talk about later. So our liver is constantly making blood proteins. We have something called albumin. We have something called fibrinogen, that's for blood clotting. We have things that are called globulins, and those are always going to be in our plasma made by the liver. So we always have protein on the inside of the blood, always in the intracellular space, but in the interstitial space, no protein. Okay, because proteins are going to be much too large to be able to fit through the plasma membrane, okay, much too large to fit through a capillary. And so our interstitial space is going to have very little to no protein, and it should be that way. If we were to have protein in our interstitial space, then it would attract water to those spaces, causing swelling in the body. So we do not want protein in our interstitial space. That is one difference between our interstitial and blood plasma. So is everybody okay with those 3 fluid compartments? Any questions? Okay. All right. When we look at a blood smear, which you looked at a blood smear back in BIOL 32, we're going to look at it again today, but now you're going to be required to know the different types of white blood cells once we get to it. Obviously, these red ones are our erythrocytes. These larger ones here, you don't need to know the names now, but I'll tell you them in lab today. And then these little pieces here, you should already know, are fragments, or platelets, or thrombocytes. And then all the white space in between is the plasma. But you'll look at those cells underneath the microscope today in lab.
Can you go back? I'm sorry.
Yeah. Now I did change the slides a little bit, mostly to keep me from writing things down and to kind of have the words already there since they're already in your lecture notes to save us time so we can make sure that we finish lecture on time. So they're going to be a little bit different. But I'll upload the most recent version because I did it late last night. But I'll upload the most recent version today. But this is all already in your notes. So, except for this one part because it doesn't mention it. But our plasma has 3 types of proteins that are produced by the liver. And those 3 proteins, the first one is albumin. Albumin is really important in our osmotic gradient or osmotic pressure. So albumin helps us to maintain our osmotic pressure. So inside of our blood, we'll have albumin that attracts water towards the plasma. Okay, so albumin attracts water towards the plasma, so we have lots of blood volume or water volume in our blood, keeping our blood pressure maintained. Okay, so it helps us maintain our osmotic pressure, which helps us maintain our blood pressure. If we have too little of that albumin, okay, what's going to happen is that you're not going to have enough water attraction towards the blood, and that water is going to fill other spaces, causing edema. So you may have a swelling in the hands and feet. That's where it's most noticeable. But when you don't have enough albumin, you don't have enough attraction of water towards the blood, and it goes into other spaces. So one thing we'll talk about when we get to the digestive system is the liver. So if you are somebody who has chronic hepatitis or maybe cirrhosis even, where your liver is not functioning properly and you're not making enough albumin, then you don't get enough water attraction towards the blood and you experience edema. And they may get swelling in the hands and feet. They may get swelling or fluid in their lungs. They may get swelling in a lot of different places. And their peritoneum is a big one. People with cirrhosis may end up with something called ascites, where they have lots of fluid in the belly that has to be drained out. And that's because of the lack of albumin, because there's not enough attraction keeping that water in the bloodstream. It gets filled up in other spaces. Another type of protein that the liver makes for the blood are globulins. Now, there are different kinds of globulins. We have like alpha, beta, gamma, but we're going to focus just on immunoglobulins. These are going to be our antibodies. And then fibrinogen is the last one. It's involved in clot formation. The way that it normally floats in our blood plasma is as fibrinogen. Fibrinogen, let's see where I'm at. Oh, that's the opposite. Let me see. Fibrin is soluble, and fibrinogen is soluble, and fibrin is insoluble. Sorry, I was making this at like 1 o'clock in the morning last night. But again, fibrinogen is involved in clot formation. So the way that it normally travels in the blood plasma is as fibrinogen, which is soluble, meaning it dissolves in the plasma. Okay, so it looks like just like the plasma, it's dissolved in there. You can't actually point it out. But when it's time to make a blood clot, we actually take fibrinogen and we convert it into fibrin. And fibrin is insoluble. So what that means is it goes from being dissolved into the blood, and then it becomes something like a piece of ice floating in water, where it comes out of the blood, where it can be used then to form a clot. So we use fibrin. Fibrin is a protein to create a mesh that forms our clot for us. Okay. And so you can consider fibrinogen as the inactive form. And then once we have an injury to our capillaries or to a vessel, and we need to form a blood clot, then we're going to turn that fibrinogen, which is inactive, into fibrin, which is active, and that will allow it to make that blood clot. Yeah?
How does it do that? Like, how does it change from fibrinogen to fibrin?
There's enzymes that will make changes to fibrinogen to convert it into that fibrin. So it's a whole process. You're not going to have to learn about it in this class, but there's like a whole like clotting cascade that involves lots of enzymes that allow for the formation of a clot. Okay, questions about those three proteins? Okay. So hematocrit is the percentage of blood that's occupied by our red blood cells. You should know that it's about 45%. So about 45% of our blood is occupied by red blood cells. Before we get into hematocrit, our red blood cell production is triggered by yet another hormone. So I'm going to give you another hormone pathway. The kidneys, and we'll talk about the whole negative feedback a little bit later, but the kidneys, when it detects things like low oxygen and knows that we need more red blood cells to carry more oxygen, it's going to release a hormone called EPO. That EPO stands for erythropoietin. And I mentioned before that for hormones only, it's okay to use an abbreviation. So if you want to use EPO, you can. But if you learn how to spell it and you can spell it correctly on a practical, I'll give you an extra 1/4 point for that as well. But the kidney, so in this case, our secreting organ is the kidney. The kidney produces the EPO, and then the EPO is going to target our red bone marrow. And once the EPO binds to the appropriate cells at the red bone marrow, the action or response then is to increase red blood cell production. So you had to make a series of flow charts for your homework today of all those hormones we learned on Monday. This is another one that you can add to that sheet. And as we go on throughout the semester, I will have you add more to it as we learn more hormones. Okay, so questions about erythropoietin. And make sure you get that right. There are going to be questions on your exam where it may ask you for the secreting organ for a hormone, or it'll ask you for the target organ of a hormone. So for this one, you want to make sure that you know the secreting organ is the kidney, while the target organ or target cells are the red bone marrow. So everyone okay with that pathway? Okay. So there is a difference in hematocrit between males and females. Females have about 5 million red blood cells per microliter, and a microliter is like smaller than a raindrop. So if you can imagine something smaller than a raindrop holding 5 million red blood cells amongst other things as well. It's pretty amazing. But we have about 5 million red blood cells per microliter. A female's hematocrit is about 37 to 47%. So about 37 to 47% of a female's blood is made up of red blood cells. And remember, the average is about 45. For males have quite a bit more red blood cells. Males have 6 million red blood cells per microliter, and their hematocrit is a little bit higher, is between 42 and 52%. The reasons why the red blood cell count is higher in males is because they have more testosterone, and testosterone is going to trigger more EPO production, and then more EPO production is going to trigger more red blood cell production. So because of their elevated levels of testosterone compared to females, they have more EPO, which means more red blood cells. So men in general are going to have thicker blood because there's more cells that are part of that blood. So they're going to have thicker blood than females. And therefore, because they've had thicker blood their entire lives, they have larger hearts than females as well. Because you need a larger heart to be able to pump a thicker, more viscous blood. Is everybody okay with that concept? Okay. And so if our hematocrit, HCT is the acronym for hematocrit. If our hematocrit is too low, so it's less than 45%, okay, our makeup of our blood is less than 45% of red blood cells, then that's going to be called anemia. So when our hematocrit drops below that 37 to 47% for females, or drops below the 42 to 52% for males, that is considered anemia. Okay? The main function of our red blood cells is to carry oxygen. So if you are anemic, meaning you have less red blood cells than you should, then you have a lower oxygen carrying capacity, which means you get less of that oxygen delivery to tissues, which means less energy for those tissues. And so because of the lowered amount of oxygen carrying capacity, the lower amount of energy that can be produced, people who have anemia experience things like fatigue, so they're tired, pallor or white pale skin. They may have low mood because of the low energy production. They may have shortness of breath. They may feel cold or chilly compared to others, and they'll have an increased heart rate and respiration rate to try to make up for that lack of oxygen carrying capacity. Yeah?
Professor, is there some truth to people that like to chew ice or bite on ice that they're anemic? Is that true? I've heard that. Is that true?
I don't know if that's really backed by science. Yeah.
All right. Thank you.
Mm-hmm. So is everyone okay with those symptoms and signs being associated with anemia? Okay.
If it's too low, severe, they'll need a blood transfusion.
Oh yeah, for sure. Yep. Now if your hematocrit is too high, so again with some med terms, A or an means without. So anemia is related to the blood, so you have less blood or less red blood cells. Polycythemia, poly meaning many, cyte meaning cells. Polycythemia is when your hematocrit is too high and you have too many red blood cells. Okay? When your hematocrit is too high and you have too many red blood cells or have polycythemia, this means that you're going to have thicker blood than usual. Which, if you develop polycythemia as an adult, that thicker blood is going to cause more strain on your heart. Because your heart's going to have to pump harder to pump that blood the same distance that it did before it was thick. And so there are a couple different types of polycythemia. We have relative polycythemia, and then we have compensatory polycythemia. Relative polycythemia is caused by dehydration or hypovolemia, meaning blood loss. And the reason for that is because if we're dehydrated and we have less water than we should, that means our blood plasma is also going to have less water. We're going to have less water all over the place. And so if we have less plasma, that means relatively we have more red blood cells. Because normally our plasma is 55% of our whole blood, and red blood cells are 45% of our whole blood. Okay? The leukocytes and platelets are less than 1%, so we don't really have to account them in this. These are averages. But plasma is 55% and red blood cells is 45%. So if we were to become dehydrated, and again, we're going to lose water all over the place. If this goes down to, let's say, 45%, then that means relatively this now is 55%. And now we have relatively more red blood cells that make up our whole blood, causing thicker blood, causing polycythemia. So does that make sense? Other times, it can be compensatory. And that is because we have tissue hypoxia. So with compensatory polycythemia, that means that our tissues are not getting enough oxygen. And so our kidneys produce more EPO to get us to produce more red blood cells to increase that oxygen carrying capacity. But then that makes our blood thicker if we're making more red blood cells than normal. But this is to compensate for the lack of oxygen. So this could be due to smoking. If you are a smoker, you get 5 to 15% less delivery of oxygen to your tissues. So if you're getting 5 to 15% less oxygen delivery to your tissues, your kidneys detect that. They produce EPO, which gets your red bone marrow to make more red blood cells. And again, if you're doing this more than normal, it's going to make your blood thicker, which is going to put more strain on your heart. Okay. Other ways to get tissue hypoxia are COPD, so chronic obstructive pulmonary disorder. So you're not getting enough ventilation, not getting enough air movement, or sleep apnea. So sleep apnea is when you briefly stop breathing overnight as you're sleeping, or you get less oxygen as you're sleeping, which can cause tissue hypoxia. If you're not getting enough oxygen as you're sleeping, you're not getting enough tissue oxygen delivery to your tissues. Again, your kidneys are going to detect that. They're going to produce the EPO, and it's going to get your red bone marrow to make more red blood cells. And again, if it's elevated, that's going to cause your blood to be thicker, putting more strain on your heart. So questions about those two? Okay. One other way to cause polycythemia that we'll talk more about later in the lecture is something called blood doping. So sometimes, and this is illegal, you can't do this in like the professional realm, but there are some athletes that may inject synthetic EPO in order to get their red bone marrow to make more red blood cells, which means they get to carry more oxygen, which means they can make more energy and maybe have more endurance during whatever sport that they're engaged in. Right? But again, that's illegal. It's performance enhancing, but definitely cannot do that. Red blood cells, talk about their characteristics. They are small and flexible. They are a biconcave shape, which means they are concave on either side. When you look at a red blood cell underneath the microscope, because it's biconcave, it's really, really thin in the middle, which is nice for us because it allows us to deliver oxygen and pick up CO2 really fast if the wall of that cell is really thin. But I'm going to go back to the picture of the microscope. You'll notice those red blood cells, they look like they're clear in the middle. They look like there's nothing in the middle. There is a wall in the middle. It's just it's so thin that the bright light from the microscope passes all the way through it. And so it looks like there's nothing there, but there is. There's a very, very thin wall there. And so this biconcave shape, this really thin wall in the middle, allows for rapid loading and unloading or diffusion of our respiratory gases. And what are our respiratory gases? CO2 and O2. Those are our respiratory gases. O2 is needed for cellular respiration, for energy. CO2 is a waste product that we breathe out. So why red blood cells are not considered a true cell? And again, I'll say red blood cells, and I'll use erythrocytes, and I'll say them both interchangeably. But make sure on an exam you need to say erythrocyte. If you say red blood cell, then it's only half credit. But these erythrocytes have no nucleus and no mitochondria, and they're actually lacking a lot of the other organelles too. The reason for that is red blood cells' primary function is to carry oxygen. So if their primary function is to carry oxygen, what they do is they get rid of all the other organelles so they have more room for more hemoglobin to be able to carry that oxygen. That's really their only job. And so we'll learn about the development of erythrocytes here in a second, but It does have a nucleus because it needs a nucleus for the DNA for the instructions to make proteins like hemoglobin. But once the red blood cell is fully developed, it actually kicks out the nucleus before it enters the bloodstream. It has no mitochondria either, so it cannot do aerobic respiration because there's no mitochondria. That's where aerobic respiration occurs. So it has to do anaerobic glycolysis instead, okay, or fermentation. And how many ATP are produced per glucose with anaerobic glycolysis? 2. Compared to aerobic, which is what? 32 to 36-ish, kind of. There's a range. It all depends on the efficiency of the individual. But it's so little compared to what we can do with oxygen, right? So only 2 ATP per glucose. The reason for that is because if the only job of the erythrocyte is to carry oxygen and deliver that oxygen to the tissues, it's not going to be using the oxygen itself. That doesn't make any sense, right? So they create their energy through an anaerobic pathway, so they're not using the oxygen that they're supposed to be delivering to other tissues. Right? Does that make sense? I always make the joke of it's like red blood cells are a drug dealer, and if you're a good drug dealer, you're not going to use your own stash. You're going to be able to sell all of it so that you make as much money as you possibly can, right? So red blood cells, they do not use that oxygen because their job is to deliver that oxygen throughout the body. And so because it has no nucleus, so no instructions to make any parts of the cell, and there's no mitochondria for no energy or very little energy that can be produced, there's no repair possible. Okay, so red blood cells cannot repair themselves, so they only live about 120 days. Okay, and you could say 100 to 120, but it's about 120 days that these red blood cells live. Okay, and again, it's mostly because of the lack of a nucleus, so they don't have the instructions. They don't know what to make or how to make it if they don't have a nucleus. So questions about the characteristics of red blood cells? Okay. So again, the function of these erythrocytes is to carry oxygen that's attached to hemoglobin and deliver that to tissues all over the body. And so without oxygen, your cells die. Why? Why do your cells die without oxygen? Can't produce ATP, right? So if you don't have energy to keep the cell running, okay, those cells die. And overall, if you lack oxygen for a certain amount of time, the entire body will die because it will run out of ATP to be able to inflate the lungs, to be able to pump the heart and do all of those vital functions that keep you alive. So when somebody dies of suffocation or asphyxiation, they're not really dying from a lack of oxygen. They're dying from a lack of ATP. They're dying from a lack of energy to keep the body running. Okay. So all of our cells are going to rely on that transport of oxygen. So how it makes that energy is through aerobic respiration. Oxygen plus glucose, which is C6H12O6, is used to make about 32 ATP, plus some CO2, plus some water. And if you want to include it, you can say that heat is produced as well. And this is another thing, you may or may not have needed to know this by heart for Bio 32, but you do need to know it by heart for Bio 33. Okay, everybody okay with the equation? Okay. So hemoglobin we're going to talk about next. Does anybody remember what kind of protein hemoglobin is? We talked about types of protein in chemistry 2 in Bio 32. There are fibrous proteins like keratin that make your hair and nails, or collagen as well. And what's the other kind of? So it's the G. Globular or globular. It is a globular protein. And by looking at the hemoglobin here, what level of protein folding do you think it's displaying? It's quaternary, yeah, because we have 1, 2, 3, 4 repeating subunits. Okay, so since we have 4 different subunits, this is showing quaternary folding. And then if it has 1, 2, 3, 4 subunits, how many molecules of oxygen do you think it can hold? 4. So it can hold 4 oxygen molecules at a time. Does anybody remember what the oxygen carrying protein is in our muscles? If it's not hemoglobin and it's muscle, what do you think it is? Myoglobin. And myoglobin can actually only hold 1 oxygen per myoglobin, while hemoglobin can hold 4. Okay. So when oxygen is bound to the hemoglobin, we call it oxyhemoglobin. This is oxygenated hemoglobin. And oxygenated blood is going to be a very bright red color. And then when oxygen is released, and there's not very much oxygen or any oxygen at all on the hemoglobin, we call it deoxyhemoglobin. And deoxygenated blood is a darker red color, almost like maroon, sometimes a purple-ish color. I would say more maroon, but it's going to be a darker red. Now, when we get into blood vessels, and even in this chapter, I may show it as well, it's going to show you oxygenated blood as red and deoxygenated blood as blue to have a more distinct color variation to tell oxygenated versus deoxygenated. But make sure that you know that blood is never, 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 never blue. It's not blue. It may maybe look a little blue or green because of the reflection and your skin and the light that passes through, but your blood is never truly blue. Okay, and I'm going to give you a hint. On an exam, I may ask you a true or false question that says deoxygenated blood is blue in color because of the lack of O2. That's false, okay? Because blood is never, 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 never blue. It goes from being bright red when it's oxygenated to a very dark red or a maroon color when it's deoxygenated. Okay? It only shows red and blue in your textbook for visual purposes. And I say that to you, and I tell this to everybody in class, and I give them the answer. People still sometimes say true, but it's false. It's going to be false. There's your 1 point on the final that I'm giving you. Okay. So we're going to talk about carbon monoxide. So CO is carbon monoxide versus carbon dioxide. So carbon monoxide is made by inefficient burning of organic matter. So like the exhaust in your car is carbon monoxide. A not very well ventilated furnace would accumulate a carbon monoxide. Your gas stoves can also leak carbon monoxide. So it's from inefficient burning of fuel or organic matter. Cigarette smoke also creates carbon monoxide. And remember, you get 5 to 15% less oxygen delivery to tissues when you do smoke cigarettes. Now, carbon monoxide is toxic at high levels, and that's why we have alarms in all of our houses for carbon monoxide. It is toxic at high levels. The reason why it's toxic at high levels is that carbon, and before we get there, carbon dioxide binds here. So hemoglobin has the ability to carry both carbon dioxide and oxygen. The carbon dioxide binds at our globin chains here, while our oxygen binds at the heme group where the iron is, that we'll talk about later. But what's so dangerous about carbon monoxide is that carbon monoxide can also bind to that heme group. So oxygen binds here, but carbon monoxide can too. And what the scary thing is that carbon monoxide has a higher affinity to that heme group on hemoglobin than even oxygen does. So it can jump on to an empty space here on the heme group, and it'll bind really tightly and not come off. Okay? So it's more attracted to that spot than even oxygen is. Okay, so our body can more easily carry that carbon monoxide than it can oxygen. So when you're exposed to high levels of carbon monoxide, that carbon monoxide jumps on those empty seats on hemoglobin, and they stay attached, and they don't really offload very easy. So at high levels, what's going to happen is carbon monoxide is going to take up all those spaces. Oxygen is not going to be able to get on, and then we're not going to be able to deliver that oxygen to our tissues. So it has a stronger affinity than oxygen, which means that we'll have a less oxygen carrying capacity if we're carrying carbon monoxide instead of oxygen. And again, it does not offload easy. It has a very high affinity. So those two things bind together very, very tightly. And so you get less O2 delivery. And so you can experience carbon monoxide poisoning. You can die from carbon monoxide poisoning because, again, that lack of oxygen, therefore a lack of ATP, then you don't have the energy to run your vital functions in the body, which, again, is why we have alarms or laws that require us to have alarms for carbon monoxide in our homes. So any questions about carbon monoxide? And that's why you should never start your car with the garage door closed. You should never want carbon monoxide to build up. Okay. All right. But no questions there? Everybody good? Okay. Hemolysis. This is the breaking down of red blood cells. So that we'll undergo hemolysis at the 120 days. But sometimes it can be what you'll learn about today in your next PLA is something called hemolytic anemia, which causes red blood cells to lyse or burst. So with hemolysis, and when that happens, it releases hemoglobin into the blood. So you can tell if someone's having a hemolytic event where their red blood cells are lysing by testing their blood for levels of hemoglobin. Because really, hemoglobin shouldn't be free floating in your blood. It should be within the red blood cells. Okay. Next, we're going to talk about blood cell production. Know that hematopoiesis is the general term for just blood cell production. So this means all blood cells. So this includes our erythrocytes, all our different types of leukocytes, and then that megakaryocyte that makes the platelets. Those are all going to be triggered to be produced by hematopoiesis. And then we'll talk about the specifically red blood cell production. We'll talk about specifically white blood cell production. But hematopoiesis is a general term for making all of our blood cells. So hematopoiesis occurs at the epiphyses of our long bones. And the reason why is because at those epiphyses we have stem cells. And remember, stem cells, you talked about like cell differentiation in Bio 32. Stem cells have not differentiated yet. So stem cells can become any one type of blood cells. A stem cell can differentiate into a red blood cell. A stem cell can differentiate into any types of white blood cells. We'll talk about eosinophils and lymphocytes and basophils. We'll talk about all those different things. So a stem cell could differentiate to any one of those types. Could differentiate into a megakaryocyte. Can differentiate into many, many, many different things. Okay? So stem cells are at those epiphyses that allows us to make those blood cells. So at an adult, hematopoiesis occurs at our red bone marrow. But in our babies and young children who don't have developed or red bone marrow, can I talk today? Red bone marrow that's developed enough, our liver and spleen will actually do the red blood cell production for babies and children until their red bone marrow is developed enough. So in adults, we typically find our red bone marrow in our epiphyses. And then what kind of marrow do we find in our diaphyses in adults? Yellow. What's yellow for? What is it for? Fat. Yeah, fat storage. So that's hematopoiesis. Then we have erythropoiesis. Erythropoiesis is specifically red blood cell production. So hematopoiesis is the production of any of our blood cells, while erythropoiesis is the production of our red blood cells, specifically our erythrocytes. Now, you don't have to know this entire pathway here. Just know that we start off with a stem cell. We call it a hematopoietic stem cell because it's for blood cell production, but it can become any one of the different blood cells. You don't need to know all the in-betweens. I won't torture you with that. The thing that you do need to know, though, is that it does have a nucleus, right? So there's a nucleus here the entire time it's developing. And then right before it becomes a developed red blood cell, it kicks out that nucleus. We don't need it anymore. So that's part of the development. So you don't need to know the names, but know that we start off with that hematopoietic stem cell. And before we become a mature erythrocyte, we must kick out that nucleus. But remember, we do need that nucleus during development so that we can do transcription and translation in order to make lots of hemoglobin. But questions about erythropoiesis. Biggest thing to know, erythropoiesis is specific to red blood cells. We start off with that hematopoietic stem cell. It develops and develops, but right before it gets released into the bloodstream, it kicks out the nucleus. And again, that's just to make more room inside the cell to be able to carry more oxygen. Okay. So this is a negative feedback loop regarding erythropoiesis and EPO that we already talked about. When someone experiences hypoxia or even pre-hypoxia, we'll talk about that when we get to the respiratory system. So they may be approaching hypoxia. Hypoxia can mean that they have a decreased red blood cell count. So if you have less red blood cells, you have less hemoglobin, and you're carrying less oxygen. Maybe you have a decreased amount of hemoglobin. So less hemoglobin means less oxygen. Or you may just have decreased availability of oxygen. We'll talk about what happens when you live in higher elevations and don't have as much access to oxygen. So if any one of those three things occur and your body experiences hypoxia or is approaching hypoxia, your kidneys are going to detect that and release erythropoietin. And remember, erythropoietin can be abbreviated to EPO. And so the kidney detects the low oxygen, produces the EPO. The EPO is going to target the red bone marrow in order to make more red blood cells. And once that happens, your oxygen levels should return back to normal because now you have more red blood cells that can carry more oxygen and deliver that oxygen to the tissues. And then your blood oxygen levels should reach homeostasis. So, and remember, testosterone, again, there's a little note in your lecture notes, testosterone triggers more EPO production, which triggers more red blood cell production, which again is why males have thicker blood and larger hearts. You can actually artificially stimulate this process. And you do that through synthetic EPO. And that's often given as a form of treatment for people with kidney disease. So if you have kidney or renal disease and you're not making enough EPO, which means that you're not making enough red blood cells to carry oxygen, you may be given an injection of EPO to try to stimulate your red bone marrow to make more red blood cells. And then we talked about blood doping as well. Again, that is illegal in professional sports in the United States, but sometimes people will inject synthetic EPO in order to get more red blood cells, carry more oxygen. That means more energy and more endurance during whatever event that they're trying to do. One way that you can naturally stimulate this is if you live in really, really high elevations, you actually have less oxygen availability. The air is thinner the higher up that you go in the mountains. And so that can actually trigger the kidneys to make EPO. It detects as there's less oxygen available than what you're used to at the ground level. And so it causes the kidneys to release EPO, which targets the red bone marrow to make more red blood cells. And then you have more oxygen to carry around. And so there are athletes, specifically like Olympic athletes, that go train like in the high mountain areas in like Colorado and stuff. And that's in order to naturally induce this process, right? It allows them to train at these higher levels, get more red blood cells, and then when they go back down to ground level to compete, now they have extra red blood cells that they're carrying around. And so they have more oxygen carrying capacity, they can make more ATP and have more endurance. Now that's legal, they can do that. That's naturally occurring. You can't keep somebody from going up in the mountains. That's different from taking synthetic EPO. It's just smart to do it that way, honestly. So you go train at high elevations, get that increase in red blood cells, and then go compete and have a little bit of an edge on your component or opponent. Okay. Questions about that? One dietary requirement, we'll talk about a couple others, and this is already in your notes. But a dietary requirement in order to make sure that we're making enough hemoglobin and red blood cells is iron. So iron is needed to make the heme group on hemoglobin that the oxygen binds to. Okay? So one of the types of anemia that you'll research about in your homework today is iron deficiency anemia. If you're not getting enough iron in your diet, then you cannot make enough hemoglobin, and therefore you cannot make enough or carry enough oxygen. And so again, the iron is needed for that heme group, and that heme group is where oxygen binds. Okay, and a couple things about iron that you should know. Iron is stored as ferritin. Okay, so we keep it in a different form. The reason why we have to convert iron into different forms is iron is pretty small. And so iron can be really quickly excreted through our urine if we kept it by itself in its original form. And we want to be able to hold on to as much iron as we can to make sure that we're making enough hemoglobin so that we can carry enough oxygen. So when it's time to recycle our red blood cells, we convert that iron into different forms so they're larger and harder to get rid of. So we store the iron as something called ferritin or hemosiderin, but you can use just ferritin, that's fine for studying purposes, but we store it as ferritin. And then when it's time to put that iron back into the blood so it can make its way to the red bone marrow so that it can be used to make more hemoglobin, we bind it to something called transferrin. And again, that's just to make the iron larger so that we don't use it in our urine accidentally. So does that make sense for everyone? And then other dietary requirements, we need to make sure we have lots of amino acids or protein in our diet. Hemoglobin is a protein, so we need amino acids to build it. B12, and you'll learn about this with your homework on anemia as well. B12 is needed for mature red blood cells. If we don't have enough B12 in our diet, our red blood cells end up being larger than they should, and they don't last as long as they should. And the folic acid is also needed for red blood cell production, which is why it's really important that folic acid is part of prenatal vitamins.
So you say folic acid is same as B12?
It's going to, I can't remember exactly what the process is, but folic acid is used to make like DNA and RNA. So we need that to make the DNA and RNA. It's going to be in the nucleus. It's going to get the instructions for the hemoglobin. Okay, now this pathway here shows red blood cell recycling. I'm going to show you another slide that kind of gives a summary so you don't have to know all the wording and all of those steps here. But what happens is after the red blood cell reaches its 100 or 120 days, and we'll say 120 days, but as soon as the red blood cell reaches at 120 days, its macrophages, is at the top here, macrophages at the spleen or liver are going to break down that red blood cell and recycle all the parts. Our body is super efficient and recycles everything, so we just break it down and then we reuse those parts for other things that we need. So the summary to it is that we take iron off that heme group and we store it as ferritin. And again, that's to make it larger so we don't excrete it. And then the remaining heme after we take the iron off is called bilirubin. That bilirubin is processed by our liver and we convert that bilirubin into bile. And then our bile is expelled through our digestive system, and it's actually the bile that makes our feces the brown color that it is. So one thing that's interesting is that if you are someone that has liver disease or cirrhosis and you're not converting that bilirubin into bile, you may end up with white feces because you do not have the bile and it's not flowing through your digestive system, and you end up with really pale looking feces. So is everyone okay with those steps? I'm sure many of you have heard of this before, but if you have a buildup of bilirubin, or actually, what's the lifespan of a red blood cell? 120 days. But if you have a buildup of bilirubin, this can cause yellow pigments to deposit in your skin, causing jaundice. Okay, and so this is common in newborns. The reason for that is when a newborn is born, you have a lot of red blood cell turnover, lots of capillary damage. So you have this little baby that's squeezing through this bony canal, right? And so it has lots of capillary damage. It loses lots of red blood cells, so it has to replenish those red blood cells and recycle the old red blood cells. So part of that process of recycling those red blood cells is to have the liver take that bilirubin and convert it into bile. But if you're an infant, your liver is like so small, and so it can't process that much bilirubin at one time, which is going to lead to the deposits of yellow pigment in the skin, causing jaundice. Yeah.
Are C-section babies less likely to develop jaundice?
There's still going to be damage there, but yeah, it'll be less likely than someone who has to go through a vaginal birth. But either way, there is going to be lots of red blood cell turnover with a newborn baby. If you're an adult, though, you can handle larger amounts of bilirubin, but you may experience some jaundice if maybe you get into maybe a traumatic kind of accident, a crushing accident that causes lots of red blood cell turnover and just lots of bilirubin for your liver to process. So you can get like temporary jaundice from that, or if you have liver disease. So if you have liver disease and your liver is not filled with enough healthy cells, then that bilirubin is going to build up, causing the yellow pigments and causing jaundice. So many people with cirrhosis will have yellowing of the skin, may even get as bad as to yellowing the eyes as well. And what else too?
Cancer.
Oh, cancer, yeah. So anything that's going to affect your liver and its ability to process that bilirubin can lead to jaundice. One thing though is one treatment that we use for newborn babies is UV rays from the lights, or Billy lights. What that does is UV rays actually change the shape of bilirubin. And one thing about bilirubin is that it's a neurotoxin. to infants. So it can be toxic to the brain. And so what UV rays do or Billy lights do, it takes that bilirubin and it changes its shape, and then it's no longer toxic. Because you learn in B32, you change the structure, you change the function, right? And so we hit those bilirubin with UV lights, it converts it to a different shape, and it's no longer harmful for the baby. And then the jaundice will dissipate. So questions about that? And look, we finished on time. Your homework, though, is on the next page after your lecture notes. Looks like this. You're just going to use your textbook or any resource that you have and make sure that it's a good resource to look up the different types of anemia. Hemorrhagic, iron deficiency, pernicious, renal, aplastic. And then there's going to be a little bit about how inherited genes can cause hemoglobin abnormalities that lead to things like hemolytic anemia. So you're going to learn about all those things on your own. That is definitely testable information for your quiz on Monday. So make sure that you don't just do it and forget it. Study it over the weekend because I will most likely have an anemia question on your quiz. Okay. Any questions? Okay, we're good.